Gynoecium. We'll discuss some points about gynoecium which you have already learned in 11th standard. Now in gynoecium we say that if only one pistil is present, it is called monocarpellary condition, and when many pistils are present in the same flower, it is called multicarpellary. Pistil and carpel are one and the same thing. Now this multicarpellary also has two divisions, that is, if the pistils are free, this condition is called as apocarpus, and when they are united or fused, we call this as syncarpus. Okay, now we'll draw the basic structure of the gynoecium. It's like that. This is the stigma portion. This will be the long style. This will be the bulging ovary part, and this is the thalamus part, the stalk of the flower. This is the ovary. This is the style, and this is the stigma. Now there are swollen parts inside the ovary which are called ovules, and these ovules are attached to the ovary wall with the help of a structure that is called placenta. There are some swollen parts inside the ovary, and these ovules are attached to the ovary wall with the help of a structure that is called as placenta. And these ovules are nothing but megasporangium. So when we talk about this megasporangium in angiosperming part, there are various types of ovules, but here we will talk about the most common one, that is the anatropous ovule. Now we will draw the structure of this ovule. This is the ovary wall. This is the structure which is the placenta. Now this will be the stalk which is running. This is the stalk, and this is the body. This is the stalk portion, and this we can call, this is the body. Now there are various layers which help in the protection of various cells inside this megasporangium, and these are called what? These are called integuments. Now two integuments are present in anatropous ovule, and hence the condition is also called as bitegument. This is the outer integument, this is the outer integument, and this is the inner integument. Now the end at which these integuments seem to be joining is called the chalazal end, and the end at which these integuments are not joining and there is a pore like formation is called the micropylar end. This is the outer integument, and this is the inner integument. Now the stalk which was used for the attachment of the body with the placenta is nothing but funicle. The stalk is called funicle. Now there is a junction between the funicle and the ovule, that is main body of the ovule, or we can say that the attachment where the funicle is joined with the body, main body of the ovule is nothing but hilum. Inside the integuments there are various cells present which are nutritive in nature, that is they are having reserve food material, and these cells are nothing but the nucellus cells. These all cells will be the nucellar cells. The female gametophyte will develop from this area. Now there is a part of the funicle which is extended or which is stressed on the main body of this ovule like this, and this is called nothing but raphe. This is called raphe. So when we are talking about a typical anatropous ovule, we see that two parts are there, one is the stalk and one is the main body. The stalk is called funicle. The attachment of the stalk with the main body is called the hilum, that is the junction between the stalk and the ovule is called the hilum. It is covered with layers which are called integuments. These are protective in nature and there are two integuments present. So this condition is also called bitegumen. This is the outer integument, this is the inner integuments, and in the center nucellar cells are present which are having a large number of reserve food material. And this is the chalazal end and this is the micropylar end. The anatropous ovule is called so because it is upside down. Now when you take any seed and when you observe that seed, like we take a bean seed and when you observe this structure, this particular structure, you will see what? You will see three markings. One will be the pore like structure which is called the micropyle. Here a scar structure will be there and here also a scar. This scar is called hilum and this scar is nothing but the raphe. You can observe this, but these structures will only be visualized in a big seed. So this is the structure of an anatropous ovule. It consists of hilum, it consists of funicle, it consists of integuments, it consists of nucellar cell, and it has two ends. One is the chalazal end and the micropylar end. Development of female gametophyte. This is the anatropous ovule. We call it as anatropous because it's upside down. We have already discussed the structure of this ovule in previous section. Now we will see what? Development of female gametophyte. Now we know that these were the cells which were the nucellar cells, and this was the chalazal end and this is the micropylar end. Now one of the cell of the nucellus near the micropylar end will develop into MMC. That is what? That is megaspore mother cell. One of the nucellar cell at the micropylar end will develop itself into MMC, that is megaspore mother cell. Now this, how we'll get the female gametophyte from this MMC? We already know that megaspores will be formed and these megaspore will result due to the meiotic division of this MMC. The ploidy of this MMC is 2N. Now this MMC will undergo what? Meiotic division, and we'll get what? We'll get 4 megaspore. So the formation of megaspore from megaspore mother cell by the meiotic division is nothing but megasporogenesis. These all 4 cells, that is megaspore, tetrad, all the 4 cells will be haploid because the megaspore mother cell has undergo what? That is the meiotic division. Now here what you have to remember, both the ends, that this is the chalazal end and this is the micropylar end. Now the 3 cells in the tetrad, this is the linear arrangement of the megaspore tetrad. Various arrangements we have already learned when we have discussed about microsporogenesis. Now the 3 megaspore will move towards the chalazal end and they will degenerate. The 3 cell, they will degenerate. Only 1 megaspore will be functional. The whole female gametophyte will be formed from 1 megaspore, hence it is also called as monosporic development. The other 3 megaspore will degenerate. When we talked about microsporogenesis, in microsporogenesis each microspore was potential pollen, but here 3 of the megaspore will degenerate and only 1 megaspore will be forming the female gametophyte, hence it is also called as monosporic development. Now after degeneration only 1 megaspore will be there. This is the chalazal end, this is the micropylar end. Now this megaspore will undergo a free nuclear division, that is nucleus karyokinesis will occur, but it will not be followed by cytokinesis. So now 3 karyokinesis will occur and we'll get what? We'll get 8 nuclei. First karyokinesis will get 2 cell. After that we'll get, this will divide, so we'll get 2 cell. Now this will divide, we'll get 2 cell. That is the 2 nuclei. After that, so after 3 karyokinesis we'll get 8 nuclei, because this is free nuclear cell wall formation will take place afterwards. So in this megaspore we can see that it is the 8 nuclei present. Now these nuclei will move towards the ends, like the first 3 nuclei will move towards the chalazal end, the other 3 will move towards the micropylar end, and 2 nuclei will remain in the center. After this, cell wall formation will occur and we'll see what? We'll observe what? 3 cells will be, 3 cells were here. 3 cells were here at the micropylar end, chalazal end, this is the micropylar, and 2 were in between. So now the cell formation will occur in the 6 nuclei. So this is the cell wall formation and 3 cells are formed here also. And these 2 nuclei will be remaining as it is. So the 3 cells which are present towards the chalazal end are called the antipodals. These 2 are the synergids. And this is what? This is the egg. And these are what? These are the polar nuclei. Now the 3 cells here, 2 synergids and one egg will be together called as egg apparatus. It will be called was what? Egg apparatus. So this is the structure of female gametophyte. It will consist what? It will consist 3 antipodals on the chalazal end, 2 synergids and 1 egg on the micropylar end, and 2 polar nuclei in the middle of the cell. So this type of development of female gametophyte is also called polygonum, and this is a very common development in angiospermic plant. Now here we can say that, see, we'll count the cell. This is 1, 2, 3, 4, 5, 6. 6 cells are there, and this is the large central cell in which the 2 polar nuclei are present. So we can say that the female gametophyte in angiospermic plant shows 8 nucleate 7 celled condition. And this is what I was talking about, that is the polygonum type of development in angiospermic plant. So let's revise. One of the nucellar cell near the micropylar end will become the MMC, that is megaspore mother cell. This megaspore mother cell will undergo the meiotic division and it will form what? It will form megaspore tetrad. Now 3 of the megaspore will degenerate and will get what? A single functional megaspore. So this development is also called as monosporic development. Now karyokinesis will occur, that is simple mitotic division. 3 mitotic division will occur, and from one cell will get, from one nuclei will get 8 nuclei. Now as we have seen that this particular mitotic division will be free nuclear, so first the nuclei will be formed and after that cell wall formation will occur. Now after this, all the nuclei will arrange themselves at their particular position in the female gametophyte. So 3 were arranged at the chalazal end, 3 at the micropylar end, and 2 remained in the center. Now cell wall formation occurs and 6 cells will be formed, out of which 3 cells at the chalazal end are the antipodals, 2 cells at the micropylar end are the synergid, and 1 is the egg which will be fertilized by the male gamete. And in the central cell there are 2 polar nuclei remaining. So we say that in the female gametophyte 7 cells are present, that is 1, 2, 3, 4, 5, 6 and 1, this is the central cell, that is 7 cells, and these 2 are polar nuclei. So 6, 7 and 8 nuclei. So we say that 8 nucleate 7 cell condition is persistent in angiospermic plants, and the egg apparatus consists of 2 synergids and 1 egg. Now these synergids will help in the guiding, or this synergid will help in guiding the pollen tube towards the micropylar end. These will be they will have that filiform apparatus which will help in this process. So this is the whole development of female gametophyte.